So Dora, thank you very much for the kind introduction. Good morning, everybody. So I guess the pressure's on me now to speak to my computer, and I try to enjoy this as much as I can. I can't see the audience. So if you have questions, use this program and write your questions, and then at the end of the presentation, I will look over them and try to answer as many as possible. So today I'm going to focus a little bit on the degradation, microbial, and also briefly I talk about abiotic degradation of vinyl chloride, organisms, pathways, and monitoring tools. Before I start, I give you a little bit of background, why it is important, why are we worried about vinyl chloride. So this is the molecule of concern, vinyl chloride. Other names are chloroethene, chloroethylene, ethylene monochloride. Here you see the structures of vinyl chloride. On the right-hand side, it's a space-filling model of vinyl chloride. In red, you see the chlorine atom. In dark color, black, grayish, the carbon atoms. And in green, the hydrogen atoms. So it's an alkene, 2 carbons, a double bond, 3 hydrogen substituents, and 1 chlorine substituent. Vinyl chloride is a gas at room temperature. It has a boiling point of minus 13 degrees centigrade. So it basically means when we, at atmospheric pressure, it's a gas, but it's relatively soluble in water. It has a solubility of 2.7 grams per liter. It's about 43 millimolar. So 43 millimoles can dissolve in 1 liter of water. So it's relatively soluble. So there are several reasons why we are concerned about vinyl chloride exposure. So vinyl chloride is toxic. There are some acute effects. That means response to high levels of vinyl chloride. It can irritate the eyes and the respiratory tract. It can inhibit blood clotting. You can get dizzy, there's drowsiness, headaches. So those are effects, acute effects when you are exposed to high concentrations. Then there are chronic effects that have been observed when you are exposed to lower concentrations, but over extended period of times. So here the first 3 bullets, these are non-cancer effects. We have liver damage, nerve damage can occur, and you may have immune reactions. Then animals experiments also demonstrated that vinyl chloride can cause reproductive developmental effects. In rats, they showed that the testicles could be damaged, and they observed decreased fetal weight in rats. So these are animal experiments. So now there's also a cancer risk. And vinyl chloride, in fact, is a proven human carcinogen. So it can cause cancer in humans, and that has been demonstrated. So there's an increased risk of liver cancer and possibly other cancers, including brain cancer. So vinyl chloride is a cancer-causing agent, and that's documented on a number of websites, including the EPA website I cite here. And there is also a publication that came out a few years ago that actually argues that the EPA guidelines are too lenient and vinyl chloride should be handled in a much more stringent way. And I thought that's a kind of an interesting discussion. So I'm citing this paper here. So how do we get exposed to vinyl chloride? So obviously people working with vinyl chloride are at high risk of exposure. So at the workplace where people are involved in the production of vinyl chloride and then the use of vinyl chloride to produce polyvinyl chloride. Polyvinyl chloride. That's the major or the only use of produced vinyl chloride, chemically produced vinyl chloride. So then obviously the handling of polyvinyl chloride products can expose you to vinyl chloride, mostly by breathing vinyl chloride. So the air containing vinyl chloride, if we breathe it, we can get exposed. And then of course with water, so the consumption of VC contaminated drinking water. That's probably the biggest interest to this group, I'm talking to today. So vinyl chloride was found in at least 616 Superfund sites of the 1,662 recognized at the time. I may not have the newest data set, but we are all aware that there are many, many more plumes of chlorinated ethenes, including vinyl chloride, that are not on the national priority list. So we probably have to deal with tens of thousands of sites where we find vinyl chloride in groundwater. So where's this vinyl chloride coming from? I already mentioned the manufacturing of vinyl chloride to produce polyvinyl chloride, PVC. Another source are accidental spills of vinyl chloride, and I give you an example. And then a third source that probably is the most significant for groundwater contamination is the reductive transformation of polychlorinated solvents like tetrachloroethene, trichloroethene, and chlorinated ethanes. And I'll talk more about this in a little bit. I also want to point out that there are natural sources of vinyl chloride and other halogenated organic material. So combustion processes like forest fires, volcanic activity, chemical processes, photochemical processes, and also biological processes can contribute to the formation of halogenated organic compounds, including vinyl chloride. And I'll give you a few more specifics in a little bit. So briefly, manufacturing of vinyl chloride. So the global production is actually quite large, about 30 million tons, and it's expected to increase at a rate of about 3% annually over the next 20 years. And the only use really is for the production of polyvinyl chloride, which is a widely used thermoplastic polymer. So you produce vinyl chloride, the VC monomer, and then in a catalytic process, you polymerize it to polyvinyl chloride, PVC. And that's the desirable product. It's used for making pipes, lots of containers, insulating material for cables, lots of other applications. Shown in the upper right is a PVC production plant in China, but there are plants like this in many industrialized countries. So this is my example for a spill of vinyl chloride that happened 2 years ago now in November 2012. It's a train that derailed over a bridge, and the derailed containers, one of them broke, and VC leaked out of this container. And approximately 23,000 gallons of vinyl chloride were released. So you see here, this is this damaged container. And I told you that vinyl chloride at room temperature is a gas. So most of this material is probably very quickly transferred to the atmosphere, where vinyl chloride is broken down by photochemical reactions. But some of this material is obviously dissolved in the water and may have caused contamination of water bodies. Well, another way to get exposed to vinyl chloride is outgassing of vinyl chloride from PVC products. And there are reports that PVC products can contain up to 1 ppm of vinyl chloride monomer, so VC. And I told you before, PVC is used in many different applications. The automobile interior, water distribution systems use mostly vinyl chloride or PVC pipes. Many toys for children are made out of PVC. And then also PVC ends up in landfills, so another source of vinyl chloride in landfills. In construction, many PVC materials are being used. Examples are shown here. And also common household commodities like shower curtains are typically made of polyvinyl chloride, PVC. So there are lots of sources, potential sources of some vinyl chloride in the air, in groundwater from the use of PVC. So then I mentioned that vinyl chloride can be generated from polychlorinated solvents. And this is an example here. We know about tetrachloroethene and trichloroethene, TCE, commonly used solvents. They were spilled, penetrated aquifers, and they can be reductively dechlorinated to, so PCE reductively dechlorinated to TCE, to the DCE isomers, trans DC, cis DC, 1,1-DCE, and those can be reductively dechlorinated to vinyl chloride. Another possible source for vinyl chloride is the degradation of PCE and TCE to here dichloroacetylene and chloroacetylene. These are abiotic degradation pathways, for example, catalyzed by zero-valent ion. And you get these get down to chloroacetylene, which can also be a source of vinyl chloride. Then chlorinated ethanes like 1,1,2,2 tetrachloroethane or 1,1,1,2 tetrachloroethane, 1,1,2 trichloroethane are precursors for vinyl chloride directly, or they are precursors to generate TCE and DCEs, which are precursors for vinyl chloride. So the point here is contamination with polychlorinated alkanes and alkenes are potential sources for vinyl chloride in groundwater. And then I told you, I mentioned briefly the natural production. I give you an example. So this is research that is becoming more and more acknowledged. In 1954, we knew about 12 naturally occurring hydrocarbons that carry a halogen substituent. In 2010, we knew more than 4,700 such compounds. I would imagine the number now is probably greater than 5,000. So the point is, there are many halogenated, naturally occurring halogenated compounds in the environment. And one of them is actually vinyl chloride. So it can be produced according to the following pathway. You have organic material, plant material, it's degraded, you get compounds like catechol, and in the presence of iron, ferric iron, you can generate benzoquinone. And in the presence of iron, chloride, oxygen, CO2, you can abiotically generate halogenated C1 and C2 and C3 compounds, including vinyl chloride. And this has been demonstrated by a number of people. So this is a possible pathway for generation of vinyl chloride naturally, so not an anthropogenic source. Now, of course, the concentrations of vinyl chloride generated in this process are very low, so they cannot explain contamination. But the point is that when you have natural processes occurring for a very long time, it means that there must be pathways and mechanisms that degrade these chemicals. If that wouldn't be the case, these chemicals would accumulate and we would find very high concentrations. That's not the case. So there must be pathways for the degradation of these naturally produced organohalogens. And these are probably mechanisms that are of interest to us that we can then exploit and use for cleanup of contaminated aquifers. So what is the environmental fate of vinyl chloride? And I put together a slide here that demonstrates proven and possible pathways that could contribute to the degradation, transformation of vinyl chloride. So you can have metabolic reduction. So organisms use vinyl chloride as a source of energy and remove the chlorine substituent and generate ethene. I talk a little more about this later. Oops. There's also the possibility of fortuitous reduction. So the organisms don't benefit from this process, but they also produce ethene. There was some talk about anaerobic oxidation of vinyl chloride. So you oxidize vinyl chloride to products like CO2 in the absence of oxygen. It is not entirely clear if this process exists and if it's relevant for vinyl chloride degradation. No organisms have been associated with this process, so it's unclear what its contribution to the fate of vinyl chloride really are. There's a possibility of abiotic degradation of vinyl chloride, and I mention that again a little later. Then you can degrade vinyl chloride by co-oxidation. So organisms, again, that fortuitously oxidize vinyl chloride and generate non-toxic products, including CO2. And you can have metabolic oxidation. So organisms that actually grow with vinyl chloride as a source of carbon and energy in the presence of oxygen. So one thing I want to point out, in all of these degradation pathways, the chlorine substituent in red here comes off as chloride, so an anion. In all these cases, it comes off as chloride, basically sodium chloride, so that's an innocuous compound. So you don't have to worry about the chlorine anymore being toxic. In all of these pathways, it comes off as chloride. So now my computer is stuck. Oh. Let's see if we can fix this. What do I do? Oh, okay. Figured it out. Sorry. To do this again, just a moment. Okay, so then in red, those are processes that occur in the absence of oxygen. So these are anoxic processes. In blue, those are processes that require oxygen. So they only occur in environments where oxygen is available. The abiotic degradation is probably more important under anoxic conditions, and I explain why later, but it may also occur under oxic conditions. Then a few comments to these different processes. So metabolic reduction. This is done by organohalide respiring bacteria, such as Staphylococcus. The key thing to remember is that these organisms, Staphylococcus, can only make a living with the chlorinated compound as a substrate to generate energy. So they require vinyl chloride to survive. And they cannot do anything else. That's all they can do. So if they see vinyl chloride, it's good food for them. It may even be the only food they have available. And they can't do many other things. They can possibly reductively dechlorinate other chlorinated compounds if available, but they can't use any other substrates. So highly specialized bacteria that, and some of them can reductively dechlorinate vinyl chloride. And we have molecular biological tools available for this process, and I mention this again later. Then for fortuitous reduction, so some microbes produce transition metal cofactors. So for example, methanogens, acetogens have enzyme systems that have coenzyme B12. And coenzyme B12 can fortuitously reduce vinyl chloride to ethene. However, the rates are very low, and it's probably an insignificant process contributing to vinyl chloride reduction to ethene. Now with the anaerobic oxidation process, we have very little information. The information that is out there suggests that this process exists, but we have no information about where this process may be important. We have no information about the microorganisms catalyzing this process. So there's a lot of uncertainty. And it's certainly worthwhile exploring in more detail if this process exists and contributes to vinyl chloride degradation. The abiotic degradation of vinyl chloride, it's a very likely process to occur, but we have very little information. Most of the abiotic work that has been done focused on polychlorinated ethenes and other polychlorinated contaminants, but relatively little is known about vinyl chloride. So this is also an area that should be explored further. The rates are probably lower, or very likely lower, than metabolic processes, but still for attenuation in large dilute plumes or in post-remediation scenarios, it may be an important contribution to natural attenuation of vinyl chloride. Now, co-oxidation has been explored in detail. It requires, the organisms doing this require a primary substrate. So, for example, methane. Methanotrophic bacteria require methane, they grow with methane and oxygen, and the methane monooxygenase can co-metabolize vinyl chloride in a fortuitous side reaction. So there's competition between the primary substrate and vinyl chloride. And the products generated, like this VC epoxide, can cause toxicity to the organism. So the rates go down. So this is difficult then to process or engineer this process so that all the vinyl chloride will be degraded. Also, you have to realize that if you implement a process like this, the primary substrate has to be available at all times, and oxygen has to be available. So from an engineering perspective, it's rather challenging to generate conditions in the subsurface to make this process work. It has been demonstrated, Perry McCarthy has done some beautiful work demonstrating how this process can be successful, but it certainly requires a lot of monitoring and process understanding. So it's not an easy process to implement. Then metabolic oxidation. So these are organisms that have that can grow with vinyl chloride. They gain energy from this process. They also generate the epoxide, but they have specific enzyme systems to take care of this, so no toxicity. And then they generate intermediates, so these organisms can grow with vinyl chloride as a source of carbon and energy. And also, it's important to know that these monooxygenases have a high affinity to oxygen. What that means, they can also operate at very low oxygen partial pressures. And we also have MBTs available to detect some of these genes encoding for these enzyme systems. I come back to this later. So now I thought a little bit about rates, and this is indicated in these pink pluses. So metabolic reduction and metabolic oxidation are probably the processes that can give you the highest rates. Metabolic reduction or organohalide respiration is an anaerobic process, so no oxygen. The metabolic oxidation is an aerobic process, so it requires oxygen. Fortuitous reduction by transition metal cofactors is very likely to be a minor contributor to ethene formation. We don't have any information about rates for abiotic degradation of vinyl chloride or anaerobic oxidation. Co-oxidation can result in appreciable rates, but again, the process is difficult to manage. So now I talk a little bit more about metabolic reductive dechlorination or organohalide respiration. So vinyl chloride is reduced to ethene. You need an electron donor for this process. And in this reductive dechlorination process, energy is generated, and this energy is captured by these organisms. That's why we call this process organohalide respiration. On the left-hand side is a scanning electron micrograph of an organism that can do this process, D. helicoccoides. Interestingly, the only bacteria that can catalyze this process belong to this genus D. helicoccoides. So there are no other organisms known that can catalyze this process. The organisms belong to D. helicoccoides, and all the strains belong to a single species that we call now D. helicoccoides mccartyi, that was recently named last year and the name, many of you will know, Perry McCarthy. It's an honor to Perry's contribution to this field of chlorinated solvent bioremediation. So all of the strains that can catalyze this process belong to the species Dihalococoides mccarthyi. So we know a little bit more about this process. We know specific genes that encode reductive dehalogenases that catalyze this process, BVCA and VCRA. And then I have a few comments here that are important to recognize. So I mentioned that some Dihalococoides mccarthyi strains can grow with vinyl chloride, but not all do. So not all Dihalococoides mccarthyi strains can grow with vinyl chloride. Now all of them that grow with vinyl chloride, they also grow with at least one of the DC isomers. Okay? Then some of these Dihalococoides strains that grow with polychlorinated ethines can co-metabolize vinyl chloride, but they cannot grow with it. And some Dihalococoides strains do not grow with chlorinated ethines at all. So what that means is just detecting Dihalococoides mccarthyi biomarkers doesn't necessarily tell you much about their ability to catalyze vinyl chloride to ethene reductive dechlorination. So that's why it's important to have specific biomarkers like BVCA and VCRA that are tightly linked to this process of vinyl chloride reductive dechlorination. If you just quantify Dihalococoides mccarthyi cells, for example, using the 16S rRNA gene as a biomarker, you may not get necessarily information about the specific activity of these organisms. So in metabolic oxidation, organisms use vinyl chloride as a source of carbon and energy. They require oxygen for this process. And some of the organisms that can do this belong to these genera Microbacterium, Pseudomonas, Nocardioides, Ochrobactrum, Ralstonia, and others. And Jim Gossett had a paper in 2010 where he very nicely demonstrated that all of these organisms possess high affinity monooxygenases, high affinity to oxygen. What it means, they can operate at oxygen concentrations of about 0.02 milligram per liter. So they are very efficient using low concentrations of oxygen. Now that's an important realization. If you think about when you look at dissolved oxygen concentrations in groundwater, many of us would say the groundwater is anoxic because the dissolved oxygen concentration is very, very low. But we do this often at concentrations of 1 or 2 milligrams per liter. So these organisms can use a much lower concentration of oxygen. What it means, we have to be very careful interpreting dissolved oxygen concentrations for decision making. So this process here in more detail, you have vinyl chloride, there's a monooxygenase, you generate this epoxide, you have a specific enzyme system, you need CoM as a cofactor, the chloride comes off, and you generate intermediates for the TCA cycle. So then I want to briefly mention abiotic transformation. So it has been demonstrated in many, many papers that iron sulfides, kinnevite, green rusts, magnetite, and probably other compounds can react with polychlorinated ethines and can lead to their degradation. So these chloroacetylenes I mentioned before are possible intermediates in this process. Now, most of these studies have looked at polychlorinated ethines, but not at vinyl chloride. So there's a knowledge gap. We know very little about these active minerals here, how they contribute to the abiotic transformation of vinyl chloride. We don't know rates, we don't know mechanisms, we don't know products. One thing to realize is that all of these abiotic reactions are intimately linked with the microbiology. What I want to say here is that all of these phases, these active minerals, are generated because of microbial activity, leading to formation of ferrous iron, leading to the formation of sulfide, so sulfate-reducing bacteria. And so this is not a joke to be mad. This is actually a concept, be mad, that we should explore in more detail. The biologically mediated abiotic degradation. Again, the point is not to look at abiotic mechanisms only. We have to look at the microbiology that generates these active minerals and then look at their interactions with contaminants like vinyl chloride. So this BMAT concept needs to be explored in more detail, especially when it comes to vinyl chloride attenuation in large dilute plumes or in post-remediation scenarios. What are the contributions of abiotic reactions to vinyl chloride attenuation? So then the last topic I want to cover are monitoring tools. You think about this, you have vinyl chloride in an aquifer, in a groundwater system, and now you want to monitor if a process works or not. So you could theoretically look at vinyl chloride concentrations. Are they declining over time? Okay, are the vinyl chloride concentrations declining? That's certainly a way of doing it. There are a few issues. For example, in many of these plumes, you also have polychlorinated ethines, and their reductive dechlorination contributes to a constant flux of vinyl chloride. So even though vinyl chloride may be degraded, the concentration may not change significantly as long as you have sources of vinyl chloride coming from upstream. Now you could also look at product formation, so concentrations of ethene. So you could look at the concentrations of ethene. But there are also some issues associated with this. Oops, going the wrong direction again. So ethene can volatilize. It's not very soluble in water. It's a gas at room temperature. Often we don't measure ethene. We measure vinyl chloride and other polychlorinated compounds, but not ethene. Then it's possible that ethene is not a conservative tracer for this process. So ethene can be transformed and degraded. And I give you an example in the next slide. And there's a possibility that ethene is coming from other sources. So it's possible that the ethene you find is not coming from vinyl chloride. Now, other products you could measure are CO2, chloride, but that doesn't really work because chloride is generally high in groundwater, so you cannot attribute changes in chloride concentration to the process. CO2 is not a marker for the process because there are many other sources of CO2 in case of metabolic oxidation. And then you could look at biomass, and that will work if the cells catalyzing the process can be directly linked to the process, but you see this doesn't work all the time. And then you can, of course, look at the catalysts directly, the enzymes or the genes, and I mentioned that in a second. So now first, ethene as a stable end product. So the point here is that ethene is not a stable end product of VC detoxification. So if you have this process, reductive dechlorination of vinyl chloride to ethene, ethene may undergo transformation to ethane. This process is typically linked to methanogenic environments, so possibly involvement of methanogens that reduce ethene to ethane. So ethene can go away and ethane is formed. Now the fate of ethane is not really clear, but this can also be degraded, most likely. Then Steve Zinder's group had a paper last year where they demonstrated ethene degradation coupled to sulfate reduction, leading to formation of CO2. Again, the point here is that ethene is not a stable end product of vinyl chloride reductive dechlorination. So don't expect to measure stoichiometric ethene at your site. So then for the metabolic oxidation process, this is work mostly done by Tim Mattis and his group at the University of Iowa. So they demonstrated, and that's not unexpected, that a 16S rRNA gene targeted approach is not helpful. And the reason is that all of these organisms that can grow with vinyl chloride, they have relatives, close relatives with similar or maybe even the same 16S rRNA gene sequence, and they cannot grow with vinyl chloride. And that can be explained by the fact that this pathway of vinyl chloride degradation is often located on a plasmid. So you can imagine an organism with a plasmid can do it, the organism without cannot. And so therefore, these 16S rRNA gene targeted approaches are not helpful. But what Tim's group has done, they developed a qPCR assay for the ETNC gene encoding a subunit for this alkene monooxygenase, this first step, and ETNE, the gene encoding for this epoxyalkane coenzyme M transferase, catalyzing the second step. So those are biomarkers for this process, and they can be quantified in groundwater. So this is a step forward. The limitation, however, is that organisms that can grow with ethene use the same genes or proteins, but not all of these ethene degraders can also grow with vinyl chloride. So there are ethene degrading bacteria using the same genes and same proteins as the vinyl chloride degraders use. So this is a limitation of this approach. But still, it can be helpful to monitor sites where vinyl chloride disappears in the presence of oxygen. So then coming back to organohalide respiration, I mentioned it before, BVCA, VCRA encode reductive dehalogenases that dechlorinate vinyl chloride to ethene. So those are obvious targets for this process, and BVCA and VCRA specific qPCR assays are available. And they have been proven to be very useful to demonstrate that this process is occurring in groundwater or in contaminated aquifers. So if you see an increase in BVCA and or VCRA over time, you can say with good confidence that reductive dechlorination is occurring. Keep in mind that these organisms can only grow at the expense of these chlorinated compounds as electron acceptors. They can't grow in any other way. So if you have vinyl chloride plumes and you see an increase in dihalococoides with these BVCA and VCRA genes, you can conclude that they must grow at the expense of vinyl chloride reductive dechlorination. In other words, if you measure biomass using 16S rRNA gene targeted qPCR assays, targeting dihalococoides genes, you can also conclude that this process is active. So this is also the next slide, indicators of ethene formation. That's also focused on dihalococoides. And so what we've done here, with the help from MI, we looked at hundreds of wells where we had information about ethene. So here in this chart on the left axis, you see ethene concentrations measured in groundwater wells. And then at the same wells, we measured total dihalococoides by quantifying the 16S rRNA genes, and we measured BVCA and VCRA using a quantitative assay. And then we built the ratio of BVCA plus VCRA, so these reductive dehalogenase genes involved in vinyl chloride reductive dechlorination, to the total number of Dihalococoides cells we quantified in these samples. And what you see is that at a ratio of 1, we see the highest ethene formation. If the ratio is lower, we see lower ethene formation, and if the ratio is higher, we don't see ethene formation. So the point here is that the Dihalococoides 16S rRNA gene to VCRA BVC ratio near unity is an indicator of ethene formation in VC plumes. Okay, so this is a good approach because we can measure the 16S rRNA gene of Dihalococoides, we can measure VCRA, we can measure BVCA, and this ratio near unity is a good indicator for ethene formation. We also noted that the ratio of Dihalococoides to total bacteria, which can also be measured using qPCR, when that ratio is greater than 0.05%, it's also a strong indicator of ethene formation. And in vinyl chloride plumes, where you see ethene formation, the Dihalococoides 16S rRNA gene abundance, if that's greater than 10 to the 6 per liter, you can assume that ethene formation is likely to occur. So these are measurable indicators that correlate with ethene formation. And then a final tool I want to present to demonstrate vinyl chloride degradation is compound-specific isotope analysis, CSIA. So the principle of this analysis is relatively simple. There's a natural abundance of C12 and C13. And the vinyl chloride now has, most of it is C12, but some of the vinyl chloride will have C13. And you can measure that. You can measure the ratio of C12 and C13 in your vinyl chloride. Oops. So now you do this at one time point, and then you come back later and do the same measurement. Now C12 is more reactive than C13. What it means is that for microbial processes, the C12 is turned over faster than the C13 vinyl chloride. So after a while, you have an enrichment in C13 because it's not converted as fast as C12. So the pool of vinyl chloride is changing. It is now enriched in C13. And that's something you can measure. So this can be measured. Specialized laboratories can measure the ratio of C12 to 13, and they can measure changes over time. So they can come up with enrichment factors of C13. And so this has been done for different processes. So for example, for aerobic metabolic VC oxidation and for VC respiration under anoxic conditions, so organohalide respiration, reductive dechlorination. The average carbon isotope enrichment factors, epsilon, for aerobic metabolic oxidation ranges from minus 5.7 to minus 8.2 per mil. For organohalide respiration, it ranges from minus 19.9 to minus 31.1 per mil. The point here is you can measure these carbon enrichment factors in your groundwater, and you can then determine, at least theoretically, if it's an aerobic metabolic process or if reductive dechlorination is occurring. And you can also demonstrate that vinyl chloride actually is being degraded because you see a change in C13 to C12 ratios. So I think the power of this tool to demonstrate degradation is great. The utility of this tool to determine which process is contributing to your degradation activity, that may be more complicated. And one of the reasons for why this is more complicated. You can imagine that you have an area where you have reductive dechlorination occurring, but maybe also some aerobic degradation in oxic pockets in the aquifer. So then you have different processes contributing to these enrichment factors. And I'm not sure if it will be possible to tease apart if different processes contribute to the overall carbon enrichment factors. So this will be very challenging. So it's a great tool to demonstrate degradation. It may not be the best tool to demonstrate which process is contributing to the degradation. Now you also have to imagine there are even more processes contributing to vinyl chloride degradation. I mentioned abiotic degradation, maybe anaerobic oxidation. They will probably have different enrichment factors. So then if you take a sample and all of these processes contribute, you will get a mixed enrichment factor, and it will be difficult to link that to a specific process. And then if you think about putting D-helicocoides to work in bioremediation, a technology that's commonly applied these days, you have your plume, you install your reactive barrier by biostimulating this area or thinking about bioaugmentation. And then you take samples and you can apply the tools I briefly discussed to measure or demonstrate that organisms, catalysts, are present and active that act on vinyl chloride. So this technology is commonly applied to achieve reductive dechlorination using D-helicocoides. Now it could theoretically also be applied to achieve aerobic vinyl chloride degradation. But that hasn't been done very often and is probably less likely to be successful. And this is my last slide. It's a cartoon, and I want to briefly explain why D-helicocoides bioaugmentation is such a promising and good tool. So these sleek looking guys on the right, those are bacteria that were isolated from the environment, transferred many times in a laboratory, and then we bring them back to the real environment, and they don't do very well. They die very quickly. Why is that? Well, they got used to these nice laboratory conditions. You know, my students and I, we take great care of our organisms, so they get spoiled. They lose genes they don't need in the laboratory, but they would need in the natural environment. We go back to the natural environment and they cannot survive. This happens to most organisms, catalyzing different processes. Now, it does not happen to D-helicocoides. Why is that? And I think the key reason here why D-helicocoides behaves differently than most other organisms is the following. D-helicocoides is a very specialized microbe. It can only do reductive dechlorination to gain energy. It can't do anything else. Many can only do a few very specific compounds like vinyl chloride. So if you take them to the laboratory, they don't have any gene to lose. If they would lose anything, they would die. Therefore, passages in the laboratory generating bioaugmentation in ocular doesn't change D-helicocoides. You can bring it back to the natural environment and it's competitive. Because it's so specialized, it can't do anything else. And that's probably a reason why reductive dechlorination can be stimulated by bioaugmentation, but these other processes like metabolic oxidation is less likely to be successfully stimulated by bioaugmentation. So this was my last slide. I want to just briefly say thank you to many of my students and collaborators. Then the main funding agencies for this work, CERDAP and ESTCP, both DoD programs. And then also my interactions with Microbial Insights, where I learned a lot, got access to data to analyze, to make these correlations. And then obviously, thank you for listening to me. So if you have any questions, I would be happy to answer. Thank you.
All right. Thank you very much, Frank. That was an excellent presentation. So let's go to the questions. Again, if you have questions, please go ahead and enter them into the GoToWebinar software. So the first question we have here is, could you elaborate on off-gassing of vinyl chloride from PVC products, particularly from water conveyance products like PVC piping? How rapid is the process and how likely might it be to be attributed to finding?
So this is not my area of expertise, but there's some literature out there that measures the rate of vinyl chloride outgassing from PVC pipes. So there's some information in the literature that gives you very precise information. I do know that there's quite a bit of variability. What company produced the PVC material, the PVC pipe? So the products of some companies contain a lower amount of vinyl chloride monomer than the products of other companies. So there's variability in the PVC itself. If you are interested, I could I give you the citations and you can maybe look in this literature yourself because I don't know the details about this. But the point is still valid that PVC pipes can increase vinyl chloride concentration in water, but probably not to an amount that would exceed the maximum concentration limit enforced by the EPA of 2 ppb. So probably there's not a lot of vinyl chloride coming out of PVC piping, but still the goal is, a maximum concentration level. Goal is 0. And so PVC piping certainly does not help to achieve that goal.
Okay, the next question. I have seen high concentrations of 1,1-DCE at some chlorinated solvent sites. Is this as a product of biodegradation, or would this likely be a component of the original chlorinated solvent source?
Okay, so 1,1-DCE is not the commonly found intermediate in the reductive dechlorination pathway. So the most common intermediate in reductive dechlorination from PC to TCE is cis-DCE. However, we do know about organisms that generate more trans-DCE than cis-DCE. So trans-DCE can be the major product of reductive dechlorination of PCE and TCE. And there's at least one publication in the literature, it was an ES&T paper several years ago, where a group demonstrated primarily 1,1-DCE formation from TCE. So 1,1-DCE can be a product of microbial activity acting on TCE. Okay, so it's possible, but it's not commonly observed.
Can you comment on any pH limitations for both aerobic and anaerobic degradation of DC?
So I have more experience with the anaerobic process. And the anaerobic reductive dechlorination process of vinyl chloride linked to dihalococoides only, right? Those are the only organisms we know of that can catalyze this process. Dihalococoides do well at circumneutral pH. If the pH is below 6, they don't do as well. They don't like that. So the rates will go down. If you reach pH 5.5, the dihalococoides that are there may still do some reductive dechlorination. But in our experiments, we could never sustain this activity at pH 5.5. In our hands, dihalococoides do not grow at pH 5.5. So the conclusion for me is that if you have pH below 6, you have to be worried about this process. Ideally, you have a process you have groundwater with pH above 6. Now for the metabolic process under oxic conditions, you have noticed probably there are many different genera that have microbes or strains that will catalyze the aerobic metabolic process, so organisms that can grow with vinyl chloride. I would assume that they have a broader range of pH optima. So it's very likely that you will find organisms that perform better at more extreme pH values, more alkaline, more acidic. But I don't know the what the limitations in terms of pH for these organisms really are. I'm not sure anybody has really looked into this. So it's possible that the pH range for the oxic process are a little greater, but I don't know for sure.
Okay, during CSIA, is the pool of vinyl chloride already enriched in the 12C since it's an intermediate in a biological pathway? Does this complicate CSIA?
Okay, so if you still have reductive dechlorination from polychlorinated ethenes occurring, this could potentially complicate the analysis. If you have a VC plume only, I think it does not make things more complicated, because anytime you go there, you have a VC plume, anytime you go there, you take a sample, you determine the ratio, and you come back later and you measure the ratio, and then you determine the change, and from that measurement, you can determine your epsilon and the extent of degradation. Now, if the pool of vinyl chloride is changing due to the presence of polychlorinated ethenes or polychlorinated ethanes, I agree with you that this is probably more complicated. Now, I'm not the carbon specific isotope analysis expert. And so this technology is being advanced very rapidly. So the experts can actually extract a lot more information than just epsilon from the data they obtain from these C12, C13 ratio measurements. And I wouldn't be surprised that in the near future, these experts will generate guidelines and information that will demonstrate that a lot more information can be extracted from these measurements. So again, I'm not the expert, but I do believe that if you have a flux of vinyl chloride coming from polychlorinated compounds, that will complicate the measurements. If you only have a vinyl chloride plume, it should be pretty straightforward. But the experts in this field can probably give you a better answer to your question.
Okay, how well does Dehalococoides do with relatively low concentrations of vinyl chloride in groundwater, such as 10 parts per billion?
Okay, so this is a good question. So you can do theoretical calculations and demonstrate what is the minimum amount Dehalococoides needs to make a living. But those measurements are a little flawed because they are based on assumptions we make, assumptions regarding the biology that we learned from E. coli. Dehalococoides works differently than E. coli, so I'm not sure how valuable those calculations are. You can also, of course, make measurements in a laboratory, grow Dehalococoides or maintain Dehalococoides at very low concentrations of 10 ppb. One of the problems we have then, we can't measure growth anymore, right? Our measurements of growth are too crude. We are not sensitive enough to demonstrate if 10 ppb can sustain growth of Dehalococoides. You have 100 cells, they increase to 200 cells, we can't measure that. My gut feeling is that low concentrations of vinyl chloride can sustain Dehalococoides. You have to realize that, I mentioned it, these organisms can't do anything else. The natural production of vinyl chloride generates very low concentrations. My hypothesis is that these low concentrations sustain the Helicoccoides populations in many different environments at very low abundance that we can't even measure. So I know this is not a satisfactory answer for you because it doesn't help you to you don't get the information and can tell people 10 ppb will work for the Helicoccoides. This is a tricky area to explore for the reasons I mentioned. But my gut feeling is that D. helicoccoides will consume vinyl chloride if the conditions are right to concentrations below 10 ppb.
Okay, can you elaborate more about the toxicity of vinyl chloride to microorganisms under aerobic conditions? Also, is vinyl chloride toxic to the Helicoccoides at any significant level?
Okay, so I'm not aware of vinyl chloride toxicity to microbes at all. I don't think vinyl chloride is toxic at concentrations that are relevant to microbes. We have not observed toxicity of vinyl chloride to D. helicoccoides, and we feed it high concentrations of vinyl chloride. Doesn't matter. They like it. For the aerobic organisms, vinyl chloride is not toxic. What is toxic is the first transformation product, the epoxide. Okay, so the epoxide, once this is formed and the organism does not have a dedicated enzyme system to transform the epoxide to something else, the epoxide is reactive and can cause toxicity to the organism. It may react with the enzyme system that forms it. In the co-metabolic process, methane monooxygenase, the epoxide can destroy the enzyme. It can react with DNA. But this only happens if the organisms don't have a dedicated enzyme to transform the epoxide. And the organisms that grow with vinyl chloride as a source of carbon and energy, they have a dedicated enzyme system to take care of this epoxide to avoid toxicity. So vinyl chloride is not toxic to microbes at the concentrations that are relevant. The epoxide can cause toxicity for those organisms that don't have a mechanism to transform it to something that's less toxic.
Okay, do we have to redefine anaerobic or anoxic with respect to DO levels, given your slide on the metabolic oxidation at very low DO levels? Or is there an overlap between oxidation and reduction processes at low DO levels?
Okay, good question. So, dissolved oxygen theoretically is a great measure. It's a good measurement. It's a good thing to know about. The problem with DO is that the measurements are not very accurate. So I don't trust DO levels that I see when groundwater is analyzed. I never trust the DO levels. The problem is that atmospheric oxygen can very quickly contaminate or change the oxygen concentration in the groundwater. And what you measure is not representative of the in situ conditions. So many of these DO measurements are flawed. That's the bottom line. Now, how to address this problem? So you can certainly use more sophisticated tools to get to the DO. And I think that would be doable. But it needs to change in the entire industry. The way we measure DO right now is not the way to do it. There must be a change. Maybe then we can trust these DO levels, these DO measurements more. If you think about reductive dechlorination, you do biostimulation, bioaugmentation, or wherever a reductive dechlorination occurs, you have an anoxic environment. This process, reductive dechlorination, it's an anaerobic process. The presence of oxygen, the helicoccoides don't do it. So this is an anaerobic process in The environment is anoxic. There's no DO. Now, at the fringes of such a reduced zone with no DO, obviously you will have some oxygen available. But again, you can't measure it. And in those zones at the fringes of an anoxic plume, where a little bit of dissolved oxygen is available, you can have metabolic oxidation of vinyl chloride occurring. But in those areas, I promise you, if you would make oxygen measurements with the current technology, you would measure a value below 2 milligrams per liter, which would be interpreted as anoxic. So it's tricky. But I do believe that in the real environment, in situ, you have an anoxic core of a plume, and in the fringes of this plume, where some oxygen, very low concentrations of oxygen are available, you may have a combination of processes, including metabolic oxidation of vinyl chloride.
Okay, the next one, nice talk. How do you explain why Dihalococoides SSU sequences can be found in environments, although no dechlorination can be stated? They can't be dead since other methods demonstrate typical looking Dihalococoides cells. So what are they doing then? Could it be novel Dihalococoides which have lost their dechlorination trait?
So it depends on what is the abundance of Dihalococoides. So if you have high abundance of Dihalococoides, you cannot explain high abundance, and let's say 10 to the 6 cells per liter. This high abundance can only be explained if the cells grew on some substrate they can utilize. And as I mentioned, they can only utilize chlorinated hydrocarbons. So there must be some chlorinated hydrocarbon around that supported their growth. Let me see your question again. So a possibility is that you have Dihalococoides growing on a chlorinated substrate that you are not aware of. And this chlorinated substrate you're looking at that is not dechlorinated is not the substrate for the Dihalococoides that are present. That is a possible explanation. I mentioned in my talk that these Dihalococoides strains that all belong to the species Dihalococoides mccartyi, they don't have the same complement of reductive dehalogenase genes. So they can do different things. So they can grow, for example, on 1,2-dichloropropane, and they increase in abundance, but they don't do anything to chlorinated ethines. Okay? So you can have different strains with different capabilities. That is a possible explanation for your observation. There may be other explanations, but I have to think more about this. So if you have data from a site where this really applies, what you describe, email me and we can discuss that in more detail.
Okay, there are a lot of questions. So what we may have to do due to time constraints is print a list of these for Dr. Loeffler and let him answer them. And then what we'll do is send the responses out to the group along with the link to the recorded version of the presentation. Again, I want to thank you for taking your time to attend the webinar today. I know your time is valuable and we appreciate that you're able to join us. So if you have additional questions, please email them to us at info@microbe.com and I'll compile a list for Frank and get those to him so that we can get the answers for you. Thank you very much.
Thank you.